Hello everyone, this is Sean Taylor, Field Application Scientist Manager for GenScript in North America. With the onset of a new surge of COVID-19 infection globally in the latter part of 2020, and new cases being uncovered of people who were infected and recovered that are becoming infected again with SARS CoV-2, the importance of measuring neutralizing antibodies in these individuals and also for vaccinated individuals over time using a high throughput and cost-effective approach has become important for us to be able to track how long those antibodies persist in these individuals to assure protection over a longer period of time. So a number of topics to go over in this talk, but I promise it will be painless and easy to understand. I'll start with the virus structure, then the mechanism of infection for SARS CoV-2, the currently available tests and the importance of timing of sampling to assure accuracy in test results. Then we'll get into the neutralizing protective antibodies and the associated tests that are currently available for using to assess those levels in individuals. Then we'll discuss a brand new test that's out there called the Cpass SARS CoV-2 Neutralizing Antibody Kit, abbreviated Cpass SVNT. We'll compare the Cpass SVNT test with traditional IgG, IgM serology tests directly, also comparing Cpass SVNT with live cell neutralization assays, and finally, a semi-quantitative approach to directly compare neutralizing antibody titers over time, which is important, as I mentioned, for vaccination and for people who have been infected and recovered from disease to assess how long those protective neutralizing antibodies can persist. And finally, as flu season approaches, obviously cross-reactivity for any test is important, and we'll go through that as well. What we have here is a cartoon image of the virus, and we have the spike glycoprotein in red and yellow here. I've colored the yellow portion of the spike protein as the receptor binding domain. So this portion of the spike protein, this is all the same protein, this red and yellow, but this portion is what binds to the host cell. So this image here, this surface here would be the host cell, and this blue patch here would be what's called the ACE2 receptor. So the ACE2 receptor at the surface of the host cell, that this could be in the lining of the lungs, as an example, permits the virus to bind by its spike protein in the receptor binding domain. So you see these two are bound here. And then the virus can enter and inject its RNA into the cell to permit the virus to replicate. So this is the way the virus is able to enter and transmit RNA to be able to replicate. So if we look at the comparison between a nucleic acid test and a serology test, the nucleic acid test is a very highly sensitive test for early detection. And this is because it's detecting the RNA while the virus is still propagating in the body. But it is dependent on sampling. So I've seen a lot of these nucleic acid tests come back negative simply because the sample wasn't taken appropriately and the RNA degraded in the sample. And if you have degraded RNA, then again the test will come back negative, and that'll be a false negative. That patient was positive, but because the sample wasn't handled properly, it could come back negative. So it's very important that sampling is done correctly and that the sample is stored in the correct buffer to assure that the RNA is preserved correctly. Now, of course, if this is done properly, this will dramatically reduce the disease period, which is the period over which an individual doesn't know that they're infected, right? Where they can go around and infect other individuals. So the sooner the individual knows that they're infected, the sooner they can quarantine and isolate themselves from other people to prevent further infection. Serology tests, on the other hand, are detecting the antibodies post-infection. So, of course, the antibodies only come about after the immune response is launched. And this is typically a week to 10 days after infection. But the antibody test could be positive when the nucleic acid test is negative, and that's because the patient has already launched an immune response, defeated the virus, and all that's left is the antibodies as opposed to the RNA. So again, this permits asymptomatic individuals to confirm post-infection, and it also permits contact tracing. So for individuals who didn't know they were infected, they can then go back and contact trace all the people they came in contact with to assure that those people remain isolated. So these are complementary tests. As I mentioned before in the previous slide, if the molecular test comes back negative, it's always a good idea to perform a serology test to just assure that that individual hasn't already been infected and has launched an immune response. So now let's get into neutralizing antibodies. So neutralizing antibodies are the antibodies that are part of the immune response, that are launched during the immune response, that can block the ability of the virus to bind to the ACE2 receptor. So these antibodies will specifically bind typically to the spike protein, and typically even very specifically to the receptor binding domain right at the tip of the spike protein to block the virus from binding to the ACE2 receptor. So here we have the lipid bilayer of a cell with the ACE2 receptor in pink, and the virus is binding here because there's no neutralizing antibodies, which permits the virus to bind, and then the RNA will be injected in the cell, and the virus will replicate. However, if a patient has already been infected, they've launched an immune response, typically they'll generate neutralizing antibodies, which will bind and block the ability of the virus to bind to the ACE2 receptor, and therefore the virus can't replicate. So you are protected from the propagation of the virus and infection. So the two most common pre-existing neutralizing antibody tests are the virus neutralization test, abbreviated VNT, and the pseudovirus-based virus neutralization test, abbreviated pVNT. Both of these tests require working with a virus, either the live SARS CoV-2 virus in the case of VNT, or a pseudovirus, which is a lentivirus typically, that's expressing just the spike protein of SARS CoV-2. So this would be an innocuous virus. In the case of the VNT, because you're working with live virus, it requires a BCL3 level containment facility, which means protective equipment, skilled technicians who work in this kind of environment with highly dangerous biomaterial, and expensive, time-consuming, laborious process. In the case of the pVNT assay, where you're working with an innocuous lentivirus that's just expressing the spike protein of SARS-CoV-2, this is a safer way of working with a live virus, and therefore doesn't require a BCL2 lab because there's no risk of infection, but it still requires working with live cells and live lentivirus, which is still time-consuming and laborious. So in both cases, you have the virus, whether it be a live virus or lentivirus, incubating with the sample, the person who's been infected previously with SARS-CoV-2, and if that sample contains neutralizing antibodies, then those antibodies, as we see here, will bind on the outside of the virus and prevent the virus from binding to the ACE2 receptor in cells that are transfected with that receptor. That's illustrated here in orange. So we can see the neutralizing antibodies prevented the virus from binding, and therefore we get no signal. In the case where we don't have neutralizing antibodies, then the virus can bind to the ACE2 receptor, as we see here, and then we get a signal. Now, there are a number of these live cell tests. In some cases, the signal is a light-emitting signal, so we see here luminescence. In other cases, it's called a plaque-reducing or foci-reducing assay, where we get more or less plaques depending on how much neutralizing antibodies are there. In all of these cases, though, for both VNT and PVNT, it's a laborious test requiring a lot of time and or a biosafety level 3 lab. In a newly invented test, we have the Cpass SARS CoV-2 neutralization antibody test. So this is a GenScript product, and this is an innocuous, easy, straightforward ELISA-based assay. Now, I'll discuss it in more detail in the next slide, but suffice to say that because it's an ELISA-based test in 96 wells, it's way higher throughput than these cell-based assays. And it only requires 1 to 2 hours to perform the test with basic reagents in a biosafety level 2 lab. So very easy test to do. It's your typical, straightforward ELISA test. So how does the Cpass SARS CoV-2 neutralization antibody test work? What we've done with this test is we've purified the components, the key components involved with docking the virus to the ACE2 receptor. So we're focusing in just here at the docking site. And what we've produced is the purified very tip of the spike protein, which is what's called the receptor binding domain. So the domain of the spike protein that locks on to the ACE2 receptor, which is depicted here in brown. And we've conjugated that receptor binding domain to horseradish peroxidase, which is what we're using as our signal generating molecule in this test. We've also purified the cytosolic domain of the ACE2 receptor of the cell, which is depicted here in green. And we've coated ELISA plates with the ACE2 receptor, as you see here, and we've produced a solution of purified receptor binding domain conjugated to horseradish peroxidase. So if we have neutralizing antibodies, which are depicted here in red, and then non-neutralizing antibodies, which are depicted here in blue, let's say from infection, we generate some of each. You'll see that the non-neutralizing antibodies, they could potentially bind to the receptor binding domain. And we'll also get a binding of neutralizing antibodies. And it's these neutralizing antibodies that will block the ability of the receptor binding domain to bind to the ACE2 receptor. If it's blocked, then we get no signal. If only non-neutralizing antibodies are in the patient's serum post-infection, then of course the receptor binding domain can bind to the ACE2 receptor, as we see here in a complex. It's a very tight complex, so this binding event is very strong, and we'll get a signal produced from the horseradish peroxidase. Now, this whole assay was invented by the Dr. Linfa Wang in his lab. The article is here. It was a preprint, and it's been published now in Nature Biotechnology. And this assay is nice because the protein components permit it to be species independent. And I'll talk about this in more detail. So this assay has been commercialized under the Cpass SARS CoV-2 neutralizing antibody kit name for CE and HSA marked products, and as the surrogate virus neutralization antibody test for RUO research use only applications. So just to give a specific example of how the Cpass SARS CoV-2 neutralizing antibody test works, what we do in this test is we start by mixing the individual sample, the person who was infected and recovered or vaccinated from SARS-CoV-2, with the purified RBD-HRP complex mixture. So we make this mixture and we incubate this mixture together for 30 minutes. After a 30-minute incubation, in the case where we're generating neutralizing and non-neutralizing antibodies, you can see that the neutralizing and non-neutralizing antibodies are binding to most of the molecules. There's one molecule here that only has a non-neutralizing antibody bound. And then after a 30-minute incubation, we add the mixture into the assay plate, which contains the ACE2 receptor coated to the bottom of the wells. So in the case of the RBD-HRP complexes that are bound to neutralizing antibodies in red, they can't bind to ACE2 receptor. This one molecule that's only bound to a non-neutralizing antibody has its receptor binding domain able to link to the ACE2 receptor, so it binds. So when we wash the plate, we wash away these three molecules because they can't bind, they're in solution, so they'll be washed out of the plate. The one molecule that could bound remains. So this is just to depict the RBD and the ACE2 receptor. So we have one molecule bound, and then we add TMB and we get a light yellow color from the HRP that's bound to the RBD. In the case where there are no neutralizing antibodies, then all of these non-neutralizing antibodies are either binding or in this case not binding to receptor binding domain. But because these are non-neutralizing antibodies, all of these receptor binding domains are now capable of binding to their respective ACE2 receptors, as we see here with the arrows. So now all four of these molecules will bind, and when we wash the plate, because it's a very strong binding interaction, they'll remain bound. And then when we add TMB, of course, there's many more HRP molecules now, which are the signal-generating molecules with TMB, so we get a darker yellow color. The more HRP, the more yellow the color. So as we have less neutralizing antibodies, we generate a darker and darker yellow color. And as we have more neutralizing antibodies, the color is much lighter here. And this can be quantified. So what are the advantages of the Cpass SVNT assay for detecting immunoglobulins? The first major advantage is that it's immunoglobulin agnostic. So this test permits the ability of detecting any class of immunoglobulin, whether these neutralizing antibodies come from IgG, IgA, IgM, or any other class of immunoglobulin. It doesn't matter. They'll bind to the receptor binding domain and block the interaction to ACE2 receptor, and we'll be able to measure this in terms of the TMB signal. So this permits an augmentation in sensitivity vis-a-vis other serology tests that are on the market. Most commercial serology tests are very specific. They use a specific secondary antibody to a particular immunoglobulin, typically IgG or IgM. So here we are able to pan everything. This is like a total neutralizing antibody measurement, which augments sensitivity of this assay. The other advantage is that it's species independent. Why is this species independent? Because these are just proteins. So if a cat or a dog or a mouse or a rat are infected with SARS CoV-2, they'll generate antibodies to the receptor binding domain, and their antibodies will bind to purified receptor binding domain in this kit and prevent binding to ACE2 receptor. So any species can be used, which makes this kit highly applicable for drug or vaccine development right from the early stages of looking at animal models or even cell-based assays, because it's species independent. So we can work from mouse to primates to humans, all with the same kit. The other advantage of using the RBD is that the RBD is highly immunogenic. It's one of the most immunogenic components of the spike protein and of the virus itself in terms of an immune response. So if a person or an animal launches an immune response, there's a very, very high probability that they will generate neutralizing antibodies to the receptor binding domain. So this was chosen very specifically as a consequence of this so that we would be able to measure the response very well. Second advantage is that the RBD is in solution. It's not coated on the surface of the plate. And in many of the serology tests that are out there, the antigen, which is what RBD is in this case, it's the antigen to these immunoglobulins, the antigen is bound to the surface of the plate in many serology tests. So serology tests use either RBD bound to the surface or the full S1 domain of spike glycoprotein or nucleocapsid protein bound to the bottom of a plate. And when you have those proteins bound, immobilized on the surface of a plate, and it's typically by a hydrophobic interaction, you can actually cause conformational changes in those molecules, which can lead to antigenic sites becoming available that would not otherwise be available in the native state molecule, which can cause other immunoglobulins to bind, and therefore cause false positive results. And this is seen a lot in these types of serology tests where you get false positives, and this is typically because of this conformational aspect of being bound to the surface of a plate. In the case of this test, the RBD HRP is in solution. It's provided in the kit in solution. So it's in a near native conformation, which forces the immunoglobulins that bind to RBD to be very specifically bind to the near native antigenic sites that would be on the virus itself, which augments the specificity. So we increase specificity, we reduce the false positive rate in this assay, and because it's immunoglobulin agnostic, we've increased the sensitivity. So this assay actually, in comparison to other traditional serology tests out there, is more sensitive and more specific. And I'll show some data in the next slides. The other final advantage of this test, obviously, is that it's a dual detection and functional assay. So this test not only has greater sensitivity and specificity than traditional serology tests, but it also tells you something about the function of the antibodies in their ability to block the binding of RBD to the ACE2 receptor, defining these antibodies as neutralizing. So this is another great advantage of this test, because neutralizing antibodies are in fact what we're really interested in when it comes to infection, because those are the protective antibodies that prevent us from having the virus propagating in our cells. And finally, it's very fast. This test is a 96-well plate-based assay that takes about an hour, a little over an hour to perform for 96 wells. And it can be automated for mass testing. So here's some data from a particular lab, Inatos Laboratories, where they compared a number of different traditional serology tests that are out there using either IgG or IgA or IgM, and compared the identical samples. They had 84 negative samples and 10 positive samples with the Cpass SVNT test that I just explained. And as we see here, all these tests gave variable levels of sensitivity and specificity, whereas Cpass was 100% in terms of both sensitivity and specificity for delineating between the negative and positive samples. But even more importantly, the positive and negative predictive value and accuracy were widely different between these assays, as you can see here, versus the Cpass test. So this is data from an independent lab that was testing the kit, that wanted to compare and assess to pick a kit that gave them the best results. And Cpass definitely showed this. In another study that we performed, we looked at, again, a number of different kits. So this was a kit that used an RBD-coated plate. This was a kit that used Spike S1-coated plates. and nucleocapsid-coated plates. And again, what we see here is in the case of the RBD-coated plate, for the 45 presumed positive samples, it called 43 of them positive and 2 of them negative. But in the case of the 23 presumed negative samples, and when I say presumed negative, these were pre-pandemic samples. They should be negative samples. It called 3 of them positive, which is a very high false positive rate. In the case of the Spike S1 coated plate, it called many of the 45 positive samples, it called many of them negative. So a lot of false negatives here. In the case of the nucleocapsid coated plate, again, it called many of the 45 positive samples negative, 17. If we go to the Cpass test, it was better than all 3 of the other tests in terms of it called most of the samples positive that were positive, 39 of the samples were called positive, and all of the pre-pandemic samples were clearly called negative. Interestingly, the 6 false negative samples from the Cpass test, the 6 samples of the 45 that were called negative here, were also called negative in the nucleocapsid test and in the S1 test, where the specificity of those tests were very good. So our suspicion is that these 6 samples were either samples that didn't seroconvert, or they were samples that were called falsely positive by qPCR. So these were presumed positive by qPCR, and there is some controversy out there with qPCR testing in terms of what the cutoff is used to call a sample positive or negative. And because these 6 samples in red were the same 6 samples that were negative in these other 2 tests, where it's not certain that these samples were in fact truly positive. And if that's the case, then again, Cpass called these samples perfectly, because these samples, if in fact were truly negative, were actually called negative by the Cpass test. In the second study, where we looked at 41 presumed positive samples and 27 pre-pandemic samples, in the case of the RBD-coated plate, it called, in this particular study, it called all the pre-pandemic samples negative, and there was 1 false negative for the positive samples. For the nucleocapsid plate, again, it called many samples negative, as it did in study 1. And for the Cpass test, again, it only called 1 sample falsely negative, and all the pre-pandemic samples were properly called negative. So again, a very good test, corroborating the 1 false negative sample was, again, in common with all these tests. So again, we're pretty sure that this negative sample either didn't seroconvert, or more likely, it was called falsely positive by qPCR. But taking all that data into account from these 2 studies, and just using the qPCR data, so we just judged everything by the qPCR presumed positive data. In terms of sensitivity and specificity, Cpass was excellent, and in terms of accuracy, positive predictive value and negative predictive value, Cpass greatly surpassed all the other tests that were out there. And the data in this table is the combined data from study 1 and study 2. So very, very nice results, corroborating the data that Inatos got from their study, actually. Now, we also compared, so basically what these results justify is the use of Cpass just in delineating between positive and negative samples versus other commercially available serology tests that are out there that just use IgG or IgM. The Cpass test clearly gives superior, more accurate results than those other tests. But now let's move to looking how Cpass compares with traditional neutralization tests. So here we compared Cpass to live cell neutralization tests that use live virus. So these would be like the gold standard tests. So using Cpass for the 66 samples we tested, we delineated 40 as being negative and 26 samples as being positive. When we look at PRNT, there are a variety of sensitivity settings that you can use in PRNT that cause the results to change. So if we look at 50% plaque reduction, 75% plaque reduction, or 90% plaque reduction, the data change. So at 50%, 2 of the samples were falsely called positive. And then at 75 and 90% neutralization, the data were very similar to the Cpass test. There was only one sample in red that was called positive in the plaque reducing test that was called negative in the Cpass test. If we go to foci reducing neutralization tests, again, what we see here with those samples is the data changes dramatically when you change the sensitivity settings with foci tests. So at 90% neutralization, many of the samples of the 45 presumed positive samples were called negative, 22 of them, almost 50-50. When we augment the sensitivity to 75% reduction in foci, then we get very similar data to the Cpass test. In this case, there was one extra negative sample called negative, but the 6 samples that were called negative in the foci test were also called negative. So these are the red samples. These red samples were the identical samples that were called negative at 75% reduction in foci. If we go to only 50% reduction in foci, it calls all the samples positive. So again, what we think here is that the Cpass test can actually be used to help calibrate some of these live cell tests, where by using Cpass, we know that at 75% reduction in foci, the results are accurate here. So it's another advantage of the Cpass assay, actually. So very nice data corroborating live cell tests with Cpass, and a message of caution as well with these live cell tests, that you have to be very careful as to which level of sensitivity is used in these tests, because depending on the level of sensitivity, the data can either change dramatically, as we see here with the foci test, or it can still change significantly with the plaque reducing test, where 2 samples became positive by using a lower sensitivity setting. So not only does the Cpass SVNT kit provide superior and more accurate data for delineating between positive and negative SARS-CoV-2 infected individuals versus traditional serology commercial tests that are out there, it can also substitute or at least complement live cell neutralization tests to determine whether samples have neutralization antibodies. But even furthermore, this test can be converted to a semi-quantitative analysis to assess neutralization antibody titers between infected individuals. And this is really important now that we're looking at vaccination, where we want to assess how the vaccine affects the titers of neutralizing antibodies generated between individuals, and what cutoff of titer offers protection versus no protection to the disease. So here we can finally use this assay to assess these differences. So the way we do this is with a neutralizing monoclonal antibody that we provide separately with the kit. And we produce a standard curve with that monoclonal neutralizing antibody, starting from 150 nanograms per mil in a 1 and 2 dilution series. And we generate the absorbance data from the kit with this standard curve, as well as the percent inhibition values, which are calculated from the quantitative instructions that we provide to people who want to run this assay semi-quantitatively. These data are then plotted on graphically in a graphical representation of OD450 versus percent inhibition, which should generate a nice linear relationship. And this is to assess where the OD value is that gives a 30% inhibition, which is the cutoff between negative and positive samples. So we just want to know this to determine what the upper limit of the quantitative range is when we're plotting our actual samples. The lower limit of the quantitative range is where the sample dilution curves start to go into their lower plateaus. So for this particular plate, the upper limit is 1.2 and the lower limit is 0.25. So what we're looking for are data for our individual samples that fall in this region between 0.25 and 1.2. And the only way to assess, to reveal points that fall in this range is to actually dilute the individual samples as well. And we recommend that samples be diluted in a 6-point dilution series, starting from a 1 in 10 dilution in the sample buffer that comes with the kit, followed by 5 1 in 3 dilutions. So from 1 in 10 all the way down to 1 in 2430. So for these 3 samples, which represent a low neutralization sample, a medium sample that contains medium levels of neutralization antibodies, and a sample from an individual that had very high neutralizing antibody titers, these are the way these curves would look for these types of samples. And we chose points from these curves that fall in the linear quantitative range, which was delineated by these 2 dashed lines. So we have, for the high sample, 0.5, medium 0.6, and an OD of 0.7 for the low. And we plot these samples, we interpolate these OD values on the standard curve. So the standard curve is plotted by the average absorbance of our duplicate absorbance values at each dilution on the y-axis versus log of the concentration on the x-axis. And we're able to get the titers from those 3 samples, which I've delineated in green, blue, and red for the low, medium, and high neutralization samples. So now we have titer values. So we calculate from these titer values. I've just tabulated them here. So we have the titer values for high, low, medium, and low neutralization. And then we multiply those titer values by the actual dilution factor that we used to get a point in the linear range. So remember, with the high titers, we had to dilute the samples a lot, all the way down to 1 in 2430 to be able to get a point in the linear range. So 23.4 nanograms per mil multiplied by 2430 is 56,862 nanograms per mil of antibody. For the medium at 0.6, that we only had to dilute the medium by 1 in 90. So we're multiplying 18.8 by 90 to generate this value. And then for the low neutralizing titer at 0.7, we only diluted that by 1 in 10. So 16.6 times 10 is only 166. And then, if you wish, you can ratiometrically assess those values by just dividing the high values by the lowest values. So the medium neutralizing sample was 10 times more than the low one, and the high one was 343 times than the low one. And we've seen this with actual samples, these kinds of ranges. So what these values mean in terms of a large sample set and in terms of can this patient with the very low titer now be reinfected? Are they susceptible to reinfection? These are all questions that can now easily be answered by using the semi-quantitative approach with the Cpass SVNT kit, as depicted here. So just to finish cross-reactivity, we're heading into influenza season, so everyone's concerned about cross-reactivity of all these kits. We've tested the Cpass kit for cross-reactivity with all the usual suspects, and the only virus that we've assessed that is cross-reactive with SVNT is SARS CoV-1, which makes this kit highly applicable even to looking at samples during influenza season, which is now upon us. And finally, just species independent, just a little bit of data. I know I explained this vis-a-vis that we're working with purified proteins, but we have tested this kit with mice, also with patient samples and with rabbits, and there have been studies using primates as well. So this test is pan-species. The kit has been regulatory approved by a variety of marks. We're still in review for FDA, so this is in process for the EUA approval, but for these other marks, the approval has been given. And if you would like more information or to learn more about the other solutions that we provide for SARS CoV-2, you can visit our website, and we have a variety of protein and antibody solutions for folks who are building their own kits or trying to use their own research or detection techniques. And finally, we have a variety of services available to help you in the production of your own assays if you wish to use these services in your research programs. So I hope this was informative, and I look forward to any questions that you might have.